Welcome again to the Eastern Sierra, the Valentine Eastern Sierra Ecological Reserve. And this is another great system to talk about root disease, Heterobasidion root disease. Now, Heterobasidion is really two different diseases in the Sierra Nevada. But before I talk about each of those diseases, let me emphasize to you something else about Heterobasidion. Two things. Number one, it is the most broadly distributed and arguably the most important native disease in the Sierra Nevada of California. Beyond that, it is arguably the most important root disease of conifer forests in the entire world. This is, with a few exceptions, Heterobasidion is a native pathogen. There are a number of species of Heterobasidion, and they have a circumpolar distribution. There are a few invasive Heterobasidion pathogens, and those are species that from North America that have been introduced into Europe, or other species throughout the world which have been introduced into the southern hemisphere, into plantations in the southern hemisphere, which are northern hemisphere species that are grown for production reasons in the southern hemisphere. So, Heterobasidion, the most important root disease globally of conifers. I think there's a good argument. If you came to me and said, I think Heterobasidion is the most important root disease, I think you have a strong argument for that. In the Sierra Nevada, it is certainly the most broadly distributed and arguably the single most important root disease of conifers. Now, as I said, in the Sierra Nevada, we really need to talk about two different Heterobasidion species and two different Heterobasidion diseases. The two that we're focused on are Heterobasidion occidentale and Heterobasidion irregulare. Now, prior to a few years ago, when it was discovered that these were actually two separate species, we tended to just talk about them as a pine type, as a fir type, and a pine type. It turns out that, and forest pathologists recognized that there were two different diseases and they had different epidemiology, but it was only recently that it was discovered that they were actually two different species. Now, that makes a lot of sense in retrospect. The epidemiology of these two diseases was different. The pathogens, the Heterobasidion pathogens that cause the diseases seem to have some different ecological dynamics. And then later, with modern molecular tools, it was shown that they were, in fact, two separate species. But the thing for you to keep in mind is that one is a disease of fir, and the other is a disease of pine, which you can see back behind me here. It's a nice lodgepole. Again, the fir disease that we want to focus on is Heterobasidion occidentale, and the pine disease is caused by Heterobasidion irregulare. And so let's go look at the epidemiology of these systems and talk about why they're different and how they're important. Let's first talk about Heterobasidion occidentale. And remember, this is the Heterobasidion disease, root disease of fir. Now, as I mentioned, our two Heterobasidion diseases have different epidemiology. And let's first talk about the fir. Heterobasidion is what's known as a root and butt rot. The pathogen can grow through the tree. It tends to grow through the tree, can infect the bole of the tree, and it will grow down, and it will attack the roots and base of the tree. Heterobasidion, all species, are white rot pathogens, or white rot fungi, meaning that they attack the lignin within the tree and leave the cellulose within. That means that the wood that gets attacked by Heterobasidion becomes spongy and is less structurally sound, and the trees eventually will fall over. Within an area that's got high recreational use, or in this case, for the Valentine Reserve, Heterobasidion can be a serious threat to public safety, and as well as structures. And there are a number of structures here on this reserve, some of them historical, and they have value for that. So as I mentioned, Heterobasidion in fir rots out the roots, causes failure of the trees. But within a stand, what Heterobasidion would do, all species of Heterobasidion will grow through the roots of the tree, and then they can infect adjacent trees. So Heterobasidion and a lot of root diseases will cause what are known as root disease centers. And the reason for this is that they can initially infect one individual, they can grow into adjacent individuals and create essentially what is a gap within the forest, a disease-driven and formed gap. All Heterobasidion pathogens, because they are good saprotrophs, once they're established in this area, inoculum can stay high enough that it will eventually reinfect trees that regenerate within the forest. So Heterobasidion gaps, both diseases that we're going to talk about, they can be persistent. So here we have a great example of a white fir. This tree is susceptible to Heterobasidion occidentale, and there are two ways that it can get infected. Fir can be infected directly by Heterobasidion. Don't really understand that process very well. It's unclear if the pathogen is infecting in some minor wounds or gaps within the bark, but we do know that fir can develop infections, and we can have root disease centers in fir without any other kinds of disturbances. Of course, I'm leading up to what those other disturbances would be. In production forestry systems, Heterobasidion can be an even more pressing problem, because the cut stumps of recently harvested trees are susceptible to infection. So, if you cut down this white fir, the exposed stump at the time that it's cut, the below ground portion of the tree is still alive, and it is susceptible to infection. And of course, you can imagine this. You cut down this tree, you've produced a massive wound, essentially a huge surface that's at risk for infection by Heterobasidion. And that's a big problem in production forestry systems, and a reason why Heterobasidion as a disease will accumulate in stands over time, and why it's such a great, why the disease overall has such great economic importance. So, fir, the epidemiology of Heterobasidion in fir, we can get infection in two ways. Heterobasidion can infect this tree directly, grow out into the surrounding trees and infect them, or if we cut down this tree, particularly if we cut down this tree at the wrong time of year, the stump can get infected, and those infections will grow through the root system and potentially infect other trees. This is especially a problem in fir, where root grafting will occur, and there's high potential for spread among individuals. Heterobasidion in pines, our second disease system. As mentioned, our two main Heterobasidion pathogens in the Sierra Nevada have different epidemiology. In pines, infection by Heterobasidion is much more dependent on the availability of cut stumps. This is a lodgepole pine here, and pine is much more resilient to infection of the bulb. So in general, as far as we know, bulb infections don't occur very often in pine. They probably can occur,
via wounding like this, because this after an initial wounding, there's that exposed living tissue, just like a cut stump. It's probably susceptible to Heterobasidion, and then outside of management systems, Heterobasidion irregulare, that's the pine type pathogen, probably persisted within forests even without the production of stumps. However, what you can kind of take as a guide for understanding how to manage these diseases, in pine, virtually all the infections that we know about, happen due to the cutting of trees. Now again, just like in the fir example, when you cut this tree, you're producing a big wound. That is a fertile ground for infection by Heterobasidion irregulare, the pine type. The pathogen will infect that stump and then grow through the roots, and then infect adjacent trees. So one of the helpful things about Heterobasidion pathology is that depending on which of these pathogens you're working with, they're very host-specific pathogens, and because of that, if you have a problem with Heterobasidion irregulare, you won't necessarily have growth of that pathogen and disease emerging in fir, because this species here is susceptible to Heterobasidion occidentale. By the same token, if you got disease in this fir, you wouldn't have the pathogen growing into your adjacent pine and infecting it and causing disease. However, that's the extent of the good news. In pine, once the pathogen is established, disease centers can grow to be very large, upwards of hundreds, sometimes 200 square meters. I have measured Heterobasidion gaps that are probably the coalescence of multiple gaps that are in excess of 10 hectares. That's a big area. That's a big open space that's been created by these pathogens. And if that is land that's being managed for production forestry, that's a lot of acreage that's been taken out of production. That could be a big problem for you if you're managing a stand for production. So again, Heterobasidion irregulare, the cutting of this stump can produce this infectable surface that's needed to create a disease center. That's how a lot of Heterobasidion irregulare gaps form, because of past management, and because it is a pretty competent saprobe. Once it's established within an area, the disease will be persistent. It's one of these kinds of diseases that we call a disease of the site. Once established, inoculum levels can remain high enough to perpetuate the disease over time. So what do we do about endemic pathogens like Heterobasidion? Well, before we talk about some stand management solutions, let's talk about some of the other processes that are happening while the pathogen may be dispersing or actually emerging as a pathogen. There's a couple of things that are true about the Sierra Nevada, challenges that are at the forefront of managing these systems that we really have to consider before we can come up with a reasonable set of management goals related to Heterobasidion. Number one, these forests are fire-adapted systems. And here at the Valentine Reserve, I've been showing you two species that couldn't be more different in terms of how they interact with fire. The two main species that we're running into here in the stand are white fir and lodgepole pine. Two species that have very different tolerances and adaptations to fire. White fir, oftentimes high levels of white fir, and we're seeing a lot of it within the stand, both in the understory and in the overstory. Oftentimes, high levels of white fir are an indication of
low levels of fire or infrequent fire or simply a long time between a long fire return interval. However, lodgepole pine is a serotinous species. It's well adapted to occasional and catastrophic wildfires. So that's what we're seeing here is a great mix of pine and white fir. That tells me that fire has not occurred in this stand for quite some time. It also says that that this stand is susceptible to both types of Heterobasidion root disease. So we might look at this stand. This is in the urban wildland interface in the town of Mammoth, and we've got a problem. This is a really great reserve. It's unique because it hasn't been logged, and yet there's an indication here that there's a high risk of potentially catastrophic wildfire. We have the right mix of species, and the species, as I look at them and read the landscape, it says to me that the wildfire has probably occurred here, dramatic wildfires potentially at different, over the course of the last couple of centuries, and it also suggests that there's definitely a risk of wildfire moving through here and impacting the town of Mammoth, which is over here. Now, as a solution to the wildfire problem, we might want to come in here and do some stand manipulation, and I think that that's a good idea. Removing some of these trees, pine and fir, in order to make a more fire resilient stand. However, remember, one of the primary ways that we develop Heterobasidion root disease is by creating cut stumps. And that's true for both disease systems. Both Occidentale and Irregulare respond to cut stumps. Even though we have this other mode of transmission into ABs, we have the risk that if we come in here and start to implement fuels treatments, thinning this stand, we could inadvertently introduce Heterobasidion root diseases into the stand, which at present doesn't really have much. So we have a management problem, which is placing a demand on us to do some manipulation in here, and we have the risk of developing Heterobasidion root disease. So how do we square this? How do we reconcile this problem? The stand needs to have some management done, but yet we have the risk that we could develop a different management problem. There are some answers to that. Heterobasidion, the good news about it is even though it is not a dispersal limited species, the spores tend to end up all over the place. They're very light. They can travel great distances. It's not dispersal limited. If you cut a stump here, you could get an infection originating from a point very far away from you. However, there is some evidence that low levels of local disease levels influence the local inoculum levels, meaning that this stand doesn't have a lot of root disease in it, and because of that, there is lower risk of getting infection. Now, contrast that with a system where there's been a lot of management in a production forestry setting. Those stands tend to have more root disease, Heterobasidion inoculum, and cutting is going to be more of an immediate threat. However, there's a way to arrest infection in cut stumps, and this is applicable both here in this Valentine Reserve example, as well as in somewhere else where there's active cutting of fir. When the stump is cut, we can actually treat it with a borate mixture. Borax is oftentimes a good compound to treat these stumps with. It's just simply boron, and what it is, it's a boron compound, and what it does is it restricts some of the surface for it restricts Heterobasidion from infecting the surface, but it does something else. It changes the ecological dynamic between Heterobasidion and other saprotrophic fungi, which are very common here within this stand. Now, as I have emphasized to you, once Heterobasidion gets established in an individual, whether it infects this AB's tree or it infects a cut stump of the adjacent
lodgepole pine and grows through the roots into the adjacent trees, we have a persistent problem. We have a disease center that can last for decades, at least decades. Some of the disease centers in the Yosemite Valley have been there for 60 years, and they show no signs of being reinvaded or having pines reestablished within them. Once this disease gets into a stand, it can be very persistent. However, if we treat a cut stump with a borate mixture, it changes the ecological dynamics and it favors saprotrophs. Now, the interesting thing about Heterobasidion is once it's established, and this is true for a lot of root diseases, once established, they are very good at holding on to territory. However, they are not very good competitors with other saprotrophic fungi, meaning that if you can change the conditions such that saprotrophs are more likely to establish on a cut stump and decompose it, you essentially take away that pathway for Heterobasidion to get established within the stand and cause disease. There's a very simple thing that you can do. Treating stumps with these borates can basically change the system and protect you from root disease. Now, think about this. With any disease, with any pest, particularly with native pathogens where we have a long evolutionary history between host and pathogen, we're probably going to have to use as many tools as we have in order to deal with the disease problem. So here, I don't see a lot of evidence in the reserve of high Heterobasidion disease levels, but I wouldn't want to introduce it. So as a manager, I might say, we should maybe consider using borates, which are environmentally pretty benign chemicals and would protect us against the risk of Heterobasidion establishment. That's both true for fir as well as pine. That might be worthwhile. They're not very expensive. They're pretty environmentally benign. They're easy to get your hands on. You can get some protection that way. So again, even though there's not a lot of disease here, you might want to avoid introducing. The other thing that we can do, and sometimes is just as effective as borates, is simply taking some soil and rubbing it on top of a cut stump. And the reason for that is because soil has high concentration, or it has saprotrophic fungi inoculum is very common within soil. And remember, what we're trying to do with borates, or really with Heterobasidion management overall, is we're trying to get saprobes into those stumps, get the saprobes established within the stumps, and Heterobasidion, because it's a poor competitor, will not be able to grow through that stump and infect surrounding healthy trees. And of course, remember, saprotrophs, by definition, are not a risk to adjacent trees. They will simply grow through the dead material, decompose it, and they take away our problem, which is this pathway of infection of stumps and Heterobasidion growing through. So borates definitely work, and there are some who argue, and even some experimental evidence for this, that rubbing the soil on top of a stump can also introduce saprotrophic fungi and protect us against emergence of root disease. The critical problem is whether that inoculum is actually in the soil or not, and that's a little bit trickier to figure out. However, if you can get the saprobes established within there, it's a very simple way. Rubbing dirt on a stump is a very simple way of limiting the disease emergence.
I thought I would end with a few photos of Heterobasidion because the Valentine Reserve doesn't actually have any Heterobasidion. It's helpful to show you what we're talking about. The disease causes a white rot, which I mentioned before, and here are some of the symptoms here in this photograph. And you can see quite clearly that the rot eventually causes the tree to fail. And here's the effect on the landscape. This is a photograph from Camp Curry in Yosemite, and what you're seeing is a map of previously documented Heterobasidion gaps, and then a coalescence of these gaps in the Camp Curry area some years later. Now, part of the reason for the expansion of these gaps is because Camp Curry is a highly developed part of Yosemite National Park, and falling trees, of course, are a great risk to public safety. Now, when you visit these stands, however, you can see that the root disease is clearly persistent within them. So some areas have been abandoned because of persistent root disease and risk to public safety. But it's still an example of once the pathogen gets established, it's pretty much set within these ecosystems, and it can continue to affect them and cause management problems.